Welcome back everyone. Today we learn about water and nutrients. Let's take a look at the functions of water. Firstly, water is a solvent for chemical reactions such as photosynthesis in plants. Secondly, water is a key component of tissues. These include cells, tissue fluid, digestive juices, and blood. Thirdly, water is essential for controlling body temperature. Water is a component of sweat, and the evaporation of sweat helps remove latent heat. Lastly, water is needed for transporting dissolved substances. These include digestive product, waste product, maintaining the turgidity of plant cells, transporting mineral salts in the xylem, and food in the phloem. We will now move on to look at carbohydrates, fats, and protein in detail. Starting off with carbohydrates. Carbohydrates are organic molecules made up of carbon, hydrogen, and oxygen. The ratio of hydrogen to oxygen is 2:1. Carbohydrates can be further classified into three groups, which are monosaccharides, disaccharides, and polysaccharides. Mono means one, thus monosaccharides are made of one simple sugar unit. Some examples of monosaccharides are glucose, fructose, and galactose. D means two, thus disaccharides are made of two sugar units. Some examples of disaccharides are maltose, sucrose, and lactose. Monosaccharides and disaccharides are simple sugar. Polysaccharide, on the other hand, is a complex sugar, and poly means many, thus polysaccharides are made of many sugars linked together. Some examples of polysaccharides are starch, cellulose, and glycogen. We will look into these three polysaccharides in greater detail later. Food that are high in carbohydrate content are rice, noodle, and bread. It is also important to know the hydrolysis and condensation of carbohydrates. Hydrolysis reaction is a reaction in which a water molecule is needed to break up complex molecules into smaller molecules. For instance, during the hydrolysis reaction of a disaccharide, we get two monosaccharides. Hydrolysis reactions are usually catalyzed by an enzyme. Condensation reaction is a chemical reaction in which two simple molecules are joined together to form a larger molecule with the removal of one water molecule. For instance, the condensation reaction of two monosaccharides gives a disaccharide plus water. Now let's look at the types of complex carbohydrates. First, we have starch. Starch is made up of several thousand glucose molecules joined together. They are the storage form of carbohydrates in plants and can be digested to provide energy for cell activities. It is found in storage organs of plants. Next, we have cellulose. Cellulose is made up of many glucose molecules joined together, but the bond between glucose molecules is different from starch. The cellulose cell wall protects plant cell from bursting or damaging, and its cellulose cannot be digested in our intestines. Cellulose is a type of dietary fiber to prevent constipation. Lastly, we have glycogen. Glycogen is made up of many branched glucose molecules. It is the storage form of carbohydrates in mammals and can be digested to produce glucose to provide energy for cell activities. It is stored in liver and muscles of mammals. There are two types of food test that can be conducted for carbohydrates. To start, we have the iodine test. This test is used to test for the presence of starch, and the procedure is as follows. Add a few drops of iodine solution. If iodine turns blue-black, starch is present. If iodine remains yellow or brown, starch is absent. Next, we have the Benedict's test. This test is used to test for the presence of reducing sugars, and sucrose is the only non-reducing sugar. The procedure is as follows. Place 2 cm³ of food sample into a test tube and add 2 cm³ of Benedict's solution to the food sample. Shake the mixture and place the tube in a boiling water bath for 2 to 3 minutes. If Benedict's solution remains blue, it indicates the absence of reducing sugar. If Benedict's solution turns green-yellow or brick red, it indicates the presence of reducing sugar. And the different color corresponds to varying levels of reducing sugar found as seen in the picture. To end of the carbohydrate segment, we will look at the function of carbohydrates. It is a substrate for respiration and the primary source of energy. It forms supporting structures, and it can be converted into other organic compounds such as amino acids and fats. It is used in the formation of nucleic acids. It is also used to synthesize lubricants such as mucus and nectar in some flowers. Moving on to fats. Fats are organic molecules made up of the elements carbon, hydrogen, and oxygen. Unlike carbohydrates, fats contain much less oxygen in proportion to hydrogen. Some sources of fat includes butter, margarine, cheese, and olive oil. In the hydrolysis of fats, fats can be broken down into simple compounds, which is 3 fatty acid molecules and 1 glycerol molecules by hydrolysis reaction in the presence of water. The hydrolysis reaction can be catalyzed by enzymes such as lipase. The food test we can carry out to test for the presence of fats is the ethanol emulsion test. On liquid food, add 2 cm³ of ethanol to a drop of liquid food in a test tube and shake mixture thoroughly. Then add 2 cm³ of water to the mixture and shake the mixture. On solid food, cut solid food into small pieces and place the pieces in a test tube and add 2 cm³ of ethanol and shake thoroughly. Allow the solid particles to settle and carefully decant the ethanol into another test tube containing 2 cm³ of water and shake the mixture. If fats are present, a cloudy white emulsion will form. The white emulsion is caused by a dispersion of minute fat liquid droplets in water. If fats are not present, the solution remains clear. To end of the section on fats, let's look at the function of fats. Fats are a source and store of energy. It is an insulating material to prevent excess heat loss, and acts as a solvent for fat-soluble vitamins and hormones. It is an essential part of protoplasm, especially in cell membranes. It can also reduce water loss from the skin surface. Lastly, we will end of this video by looking at proteins. Proteins are organic molecules made up of the elements carbon, hydrogen, oxygen and nitrogen, and another element sulfur may also be present. Every protein molecule is made up of amino acids, and the structure of amino acid is made up of the presence of an amine group and carboxylic acid group. There are 20 naturally occurring amino acids which has different or group of atoms as seen in the diagram here. Some sources of proteins include meat, fish and egg white. The hydrolysis and condensation of proteins is as such. In the condensation of proteins, water is released as a byproduct. Amino acids, being the basic unit of proteins, can be condensed to form peptides and then further condensed to form proteins. In most proteins, the polypeptide chains are folded into more complex, 3D shape. In the hydrolysis of proteins, it is the reverse of condensation for digestion purposes. Enzymes such as peptidase and water are needed for hydrolysis to occur. The food test for proteins is the biurate test. The biurate solution is a blue solution made up of sodium hydroxide and copper 2 solution. Add 2 cm³ of sodium hydroxide to 2 cm³ of food solution in a test tube and shake it thoroughly. Then add 1% copper 2 sulfate solution, drop by drop shaking after every drop. If solution turns violet, proteins are present. If solution remains blue, proteins are absent. To end this video, let's look at the function of proteins. It is used in the synthesis of new protoplasm, for growth and repair of worn-out body cells. It is also used in the synthesis of enzymes and some hormones. Additionally, proteins are used in the formation of antibodies to combat diseases. That's all for today. Thank you for the effort to learn with me. Stay tuned for more interesting lessons. See you next time. Bye-bye.